And here we are, Protein Data Bank at 50 years of age. I have been at Rutgers since 2013 and been very much enjoying the opportunity to contribute in many different ways to the organization and to the Protein Data Bank. At the US-funded RCSB Protein Data Bank, we're turning global data into global knowledge, which entails taking the 3D structures that are coming from more than 50,000 structural biologists, PDB depositors worldwide, bringing them into the archive in collaboration with our worldwide Protein Data Bank partners involving deposition, validation, biocuration, and remediation. And then we are managing the archive, as the designated WWPDB archive keeper, making the information available to our partners and more than 400 different scientific resources. Our data exploration team delivers the same information to millions of users around the world on an annual basis through our rcsb.org website that I mentioned earlier. And then our education and outreach team, led by Christine Zardecki, my deputy director, delivers content of interest to both educators and students on pdb101.rcsb.org. And last year, the number of unique users exceeded 850,000. A recent paper in nucleic acids research describes the activities of the organization. The PDB has had impact over the last 50 years on fundamental biology, and the particular area of science that we've highlighted in this slide concerns our understanding of DNA packaging in 3 dimensions. The publication describing the nucleosome core particle, Luger et al. Has been cited more than 10,000 times. This is the most highly cited PDB structure, and it's one of more than 300 PDB structures that help us understand how DNA packaging works inside eukaryotic cells. We have DOE funders and are mindful of the impact that we're having on energy research. And you can see here in the summary that there are more than 400 PDB structures that help explain in 3D how photosynthesis works. And these structures are guiding research into how it can be harnessed by man with the goal of addressing the slow motion public health emergency that is climate change, which is obviously front and center this week in Glasgow. Impact on biomedicine's been another area where the PDB structures are influencing the outcomes of research and development efforts to the betterment of all humanity. And these next 2 slides summarize the fact that of the 210 new drugs that were approved by US FDA 2010 through 2016, discovery and development of 184 of these 210 were facilitated by nearly 6,000 structures that were available in the PDB, open access with no limitation on usage. A similar analysis that was carried out looking at the approval by US FDA of 79 new anti-cancer drugs between 2010 and 2018 documented that 74 of these 79 new anti-cancer drugs had their discovery and development facilitated by nearly 2,500 structures that were in the PDB. And importantly, 54 of these new drugs targets of 54 of these new drugs were small molecules, and 70% of those small molecule drugs were the product of structure-guided drug discovery. In many cases, the starting point for the discovery effort came from an existing PDB structure as it was carried forward in industry. We've been living the pandemic, and PDB has been very important in helping to accelerate research in the fight against SARS-CoV-2. As of today, there are 1,558 SARS-CoV-2 related structures in the PDB. The very first one was actually deposited in January, that of the main protease, PDB-ID 6' uniform 7. We've also done a very considerable amount of outreach. Much of it has been motivated by the molecular art that our artist in residence, research professor here at Rutgers, David Goodsell, has produced. And this is the unseen enemy. This is a rendition of SARS-CoV-2 inside an aerosol droplet. And this is why we have to wear masks and socially distance until we have enough people vaccinated or otherwise immune to infection as a result of previous exposure to the virus to actually ultimately tamp down the pandemic. One of the most important things that's happened over this last 12 months is that in structural biology is the fact that protein structure prediction has come of age. We have seen over many years now a series of major milestones which have culminated in the successful application of artificial intelligence machine learning tools to the problem of protein structure prediction. And I'm of course referring to AlphaFold DB and Rosetta Fold. And I think it's fair to say that that both Google DeepMind and the Rosetta team have vindicated the statement made by Sir Isaac Newton: if I have seen further than others, it is by standing on the shoulders of giants. And the contributing infrastructure here has been very significant. Open access to PDB data was key, access to deep genomic sequence data, a series of challenges that have been carried out both episodically and weekly, sharing of information through peer-reviewed publications, and sharing of computer code via GitHub. All of these things came together to make it possible for DeepMind and the Rosetta team to enjoy the successes that they have with the machine learning methods. I want to present a brief case study here to highlight some of the strengths and potential weaknesses of de novo protein structure prediction using AI. This is the human Tweety Homolog 2. AlphaFold 2 was used to predict the full-length structure, which is depicted here with its code from the AlphaFold DB. It's a mostly globular protein with high confidence in the blue regions, and then the C-terminal 130 residues were predicted by AlphaFold with either low or very low confidence, meaning that you cannot believe the atomic coordinates of these lightly colored parts of the structure. This computed structure model was generated before PDB-ID 7 Peter 5.4 was deposited to the PDB. You can see the structure here. It's a symmetric homodimer with 3 glycosylation sites per monomer. It's an integral membrane protein, and the C-terminal 130 residues or so was not actually visible in the cryo-EM map, reflecting the fact that it's probably intrinsically disordered. When the experimental structure determined by electron microscopy was compared with that of the AlphaFold DB computed structure model, the root mean square deviation was an impressive 0.8 angstroms. So as we look ahead and think about the impact that the PDB is going to continue having, and particularly the amazing amount of 3D structural information that's now become available for proteins. I'm going to make the statement that I believe that reliable 3D biostructure data, no matter where it's coming from, will enable research and education across the natural and engineering sciences and across medicine. The challenge for us is going to be integrating the information that's available from the PDB, the experimental structures, with those of computed structure models coming from a very large number of sources, and making this corpus of data readily accessible and understandable to biologists, engineers, technologists, clinicians, etc. To this end, on the 9th of December, next month, we're going to do another Institute crash course, again virtual, that will be entitled Enabling Protein Structure Prediction with Artificial Intelligence at Rutgers and beyond. There'll be some didactic lectures at the beginning of the afternoon session on Thursday, December the 9th, followed by a hands-on tutorial during which participants will have the opportunity to actually compute, either download pre-computed or to compute structure models for their protein of interest, assuming that it's not present in the protein data bank. So I urge all of you to register. The information is available on the IQB website, iqb.rutgers.edu, and the registration link here will make it possible for you to register. If you do have a Rutgers ID, provide it during the registration, because that will then give you an entree to the Amarillo supercomputer cluster that's being made available for the crash course. This is being put on in collaboration with the Office of Advanced Research Computing, led by Bar Van Osen, and my co-organizer on this crash course is Bala Desingyu, who's probably well known to quite a few of you. So looking further ahead, as we think about the impact of computed structure models, one of the I'm going to make three statements and then discuss two challenges. Appropriate use of computed structure models can advance functional understanding even before experimental structures become available. And I think you could see that in the case of the Tweety homolog, that there was the quality of the prediction that for the globular part of the structure that came from AlphaFold would have been sufficient to begin planning functional experiments. Computed structure models are also going to accelerate hypothesis generation and structure determination, particularly expression construct design for functional studies in 3D using EM, NMR, X-ray, tomography, etc. And the other area where I think computed structure models are going to contribute significantly is that it will accelerate progress in so-called integrative or hybrid methods structure determination. At present, these so-called IHM structures are going into the PDB dev prototype archive, but our goal is to eventually fold those into the PDB and to support deposition of IHM structures through the Worldwide Protein Data Bank deposition system. There are some challenges as we think about how we're going to use computed structure models alongside PDB structures. The first is the need for interoperable data standards, and fortunately we're in very good shape here. I mentioned the PDBX SIF data standard that was developed by John Westbrook while he was a graduate student here at Rutgers. Since PDBX MMSIF became the data standard for the PDB, it has also been adopted by some members of the modeling community and is now there's now a model SIF which interoperates with PDBX MMSIF. The AlphaFold DB models are being provided in model SIF, as are the Rosetta Fold models. The other thing that's going to be required to be able to readily work with both computed structure models and experimental structures will be a common validation framework that will work across both types of structures, those structural models, those coming from experimental, those coming entirely from computed models. And we believe that Bayesian methods are going to be the key here, and the Worldwide Protein Data Bank is currently working to assemble a team of experts from the community, just as they did with validation, to advance this aspect of the challenge. And so with that, I want to say thank you, and I want to acknowledge that I'm only able to stand at the tip of this spear as a result of the efforts of a very large number of individuals who are contributing to the RCSB PDB team at Rutgers, UCSD, and UCSF, funded by NSF, various NIH institutes, and the US Department of Energy. All of this, of course, is happening in partnership with our Worldwide Protein Data Bank collaborators. And I will close with a job ad. We are currently seeking scientific software developers, both at Rutgers and UCSD. We have an opening for a postdoctoral researcher expert in computational bioinformatics at UCSD. And we're also seeking the services of a cloud computing sysadmin here at Rutgers.